Hey everyone, welcome back. So now we are continuing after having studied esophagus. We're going to be talking about the stomach and the intestines. So in the intestines, we'll talk about the small intestine in today's video along with the stomach. So let's begin. The stomach is a J-shaped organ, although it doesn't really look like a J. The esophagus connects it from the top and from the bottom. The tube is continued with the small intestine, particularly the part of the duodenum is coming out of the stomach from the lower end. The upper side of the stomach is known as the cardiac region because it is very close to the heart. And then the main region where the digestion happens is the fundus, and then the lower part, which is connected, is known as pyloric region. If we cut the stomach, we are able to see that there are various layers inside the stomach. The most important of this is the muscularis, like we talked in esophagus also. The muscularis also here has the longitudinal and the circular muscles. Along with these two, there's also a layer of the oblique muscles, but we don't have to talk about it. When we divide the function of stomach, we can categorize it into two parts, namely the mechanical digestion part, where there's no chemical change, but there is physical breakdown of food, and then there is a chemical digestion part, where a chemical reaction happens to break big molecules into small soluble molecules. The mechanical digestion is favored by peristalsis. We know peristalsis, the rhythmic wave-like motion, which allows the churning of food within the stomach. Now, by this churning, we refer to the fact that this makes a paste of food known as chyme, Chyme. I don't know why I wrote A, but it should be E. So this chyme is known as the food paste made by the churning, so provide high surface area for better enzymatic action because it's easier for the food to act over paste. In the chemical digestion part, the first important thing is HCl, and the second thing that we go and find in the stomach is the pepsinogen enzyme. HCl stands for hydrochloric acid. If you take chemistry, you would know it's a strong acid. So HCl kills the pathogens because it's a really acidic environment and all the bacteria are dead. HCl also activates the pepsinogen because pepsinogen is the inactive form of the enzyme. HCl also provides the optimum pH, by the way. Pepsinogen is activated to form pepsin, which is a kind of protease. So proteins are broken down by the help of this enzyme into peptides. So in the presence of acidic medium, this reaction happens. Now moving on, let's talk a little about the small intestine. The small intestine, as you can see in the diagram, originates directly from the stomach. I have made it really thicker. It's not as thick, but for the sake of the clarity. So the small intestine, starting from the stomach, is divided into three parts. The first part is known as the duodenum. Then there's a middle part, which is known as the jejunum. And then after the jejunum, the final part is known as the ileum. Digestion happens in duodenum and jejunum with the help of liver and pancreas as supporting organs. Liver has a gallbladder which stores a chemical known as bile. And then pancreas is near the stomach, which has a duct known as the pancreatic duct. From the liver, the bile duct comes into the intestine, which is known as the hepatic duct, and then from the pancreas you get the pancreatic duct. These ducts enter the duodenum part and bring the important chemicals. The bile duct brings the bile, and pancreatic duct brings the pancreatic juice, which includes pancreatic lipase and pancreatic amylase. You can figure the function of these enzymes already. When we talk about the food passing through the duodenum, the bile and the pancreatic juice help in the digestion of starch and lipids. At the same time, the secretory cells, the cells of the jejunum itself, produce a lot of enzymes. For example, the intestine produce enzymes like maltase, sucrase, lactase, and peptidases, etc. We'll talk about it shortly. So these enzymes are produced by the intestine itself, but there are something produced by the pancreas also. Let's quickly write the theory down so we have some written content with us. So first let's talk about the duodenum as a revision. In the duodenum, the liver produces bile which was stored in the gallbladder. Bile is a green-brown colored chemical. It helps in the emulsification of fats, or you can say the emulsification of lipids. So the lipase, which is an enzyme, so the lipase can digest it. Because fats are water insoluble, right? So we need the emulsification. So let's write it. Since lipids are insoluble in water, that is why we need emulsification to mix it with water along with the enzymes. That is what bile does. What does pancreas do? Pancreas produces a pancreatic juice which has the amylase enzyme. So starch can be broken down into maltose. Starch is a polysaccharide, maltose is a disaccharide. It also has lipase, which breaks down lipids into fatty acids and glycerol. We already know that. Both of these are hydrolysis reactions. When we talk about the jejunum, it has enzymes mainly produced by the intestine itself, like trypsin, which is a kind of protease. Protease means the proteins are broken down by trypsin into peptides. Then we got peptidases, because peptides are also not the smallest of the molecules. Even peptides can be broken down. So peptidases break down peptides into amino acids. Amino acids are the smallest possible molecule. Then what we have is maltase. We can figure out what it does. It breaks down maltose into 2 glucose monomers. And glucose is the smallest of the monosaccharides. Then we got lactase. Lactase breaks down lactose sugar into glucose and galactose monomers. Then we got finally sucrase, which breaks down sucrose, which is again a disaccharide, into a glucose monomer and a fructose monomer. This is how almost all the molecules are digested. If we enlarge ileum and we try to talk about absorption process, the ileum has this brush border like structure on the inside. The inner side, the inner opening is known as the lumen, which has this brush border like structure. These brush border like structures are known as the villi. VILLI. Now as we look closer, we know that there's an artery coming towards the intestine, obviously from the heart, which brings oxygenated blood, and there's a vein going back. That means there's a constant supply of blood. There are blood vessels within each villi, and as these food molecules, which are now digested food substances like glucose molecules and other monosaccharides, for example, all the kinds of amino acids which are now digested and ready to be absorbed, ions and water, all these substances, as they are moving through the small intestine ileum, the brush border like structures are ready to absorb them because they got capillaries. These red and blue things that you see are capillaries, and this wall of the brush border villi are made up of epithelial cells known as microvilli. We will talk about the shape of the microvilli soon. I haven't made them here, but you can see that it has capillaries and the surface of epithelial cells, which helps in the absorption of the food. And this food, once it's absorbed into the capillaries by the process of diffusion, which means from higher to lower concentration gradient, also active transport, which requires energy, and then water is absorbed by the process of osmosis. So once all these food substances are absorbed into the blood capillaries, or you can say the lymphatic vessels, it goes into the blood vessel known as hepatic portal vein towards the liver. So all the food absorbed in the ileum goes through the hepatic portal vein towards the liver, because the liver is in charge of deciding what remains and what goes out of the body. Now you can see the structure again of the villi. You can see the lumen of the intestine, where the digested food is passing through in a direction from left to right. It includes the glucose, amino acids, other monosaccharides, ions, vitamin C because that's water soluble, water. All these substances are passing through the ileum. And you can see the surface of each villi, I should say each villus because that's for singular. Each villus has many cells known as microvilli. They have these extensions. They have a very similar shape to root hair cell to increase surface area. So a villi has many microvilli on its surface, and the blood capillaries are on the inside where food is absorbed. So blood absorbs all the water soluble substances and then makes it a part of the blood, where later it goes towards the liver. So let's write some things down so that you can study and prepare for your examination for a theoretical answer point of view. Villi provides high surface area. Then you can say that microvilli, which are the epithelial cells, you should know that microvilli are cells, they are living cells. So microvilli aids in more surface area, number one, for more efficient absorption process. And by more efficient absorption, I mean more efficient diffusion and active transport. Then you can talk about the fact that there are blood vessels, which means blood capillaries, inside each villus to absorb the digested food substances, like all the substances I've named already. Then you can say these yellow blood vessels are known as lacteals, or you can say lymphatic vessels. They absorb the fatty acids because fatty acids are water insoluble. So lymphatic vessel helps in the absorption of fatty acids. And ultimately, fatty acids also become a part of the body by the absorption in villus. Overall, all these food substances are digested into the blood vessel, or you can say the lymphatic vessel, and then ultimately all the food, once it's absorbed from the entire, you can say, villi, it becomes a part of the hepatic portal vein. And then hepatic portal vein takes all the digested food substances towards the liver, where liver will decide what stays in the body and what should not, and rather should be eliminated. I hope this picture is clear for you guys. In the next video, we are going to talk about the liver indeed. Stay tuned guys. Thank